the fungi group, we have studied the general characteristics, the type of thallus, reproduction, and classification. So in the continuation of this fungi, we shall study about one typical example coming under class Phycomycetes. So already we have studied the classification. Fungi are divided into Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. So Rhizopus stolonifer is one example which we have to study about its morphology, structure, and reproduction. So Rhizopus, commonly called as the bread mold. So this Rhizopus stolonifer, it is a common fungus in where everybody has seen in our house, and it is growing usually on foodstuffs and various kind of items. It may be a fruit or it may be any kind of food which we can find this cottony mass, fine thread-like structures made up of hyphae. Already we have seen the fungi. The body of the fungi is made up of hyphae, which are fine thread-like structures. The hyphae constitutes the mycelium. So similarly, Rhizopus is one such bread mold consisting of fine thread-like structures. So this Rhizopus commonly grows on fruits, vegetables, and any other food materials, and on a decaying material. That means when we keep a fruit or any vegetable or even any food material for a long period without unnoticed, means which we do not be attentive about that, and there a kind of cotton-like mass, a cottony mass appears on the surface, and when we closely observe that structure, it consists of fine thread-like structures called as hyphae. That hyphae constitutes the mycelium. So that is the occurrence of the Rhizopus. And when we come to the thallus of Rhizopus, so Rhizopus consists of hyphae, that is aseptate hyphae. So in fungi, Phycomycetes consists of aseptate hyphae. The remaining groups, they are made up of septate hyphae. So this Rhizopus consists of hyphae, and this hyphae is having the nuclei freely present in the hyphae like this. So there are no cross walls. In fact, if the cross walls are present, they are slightly present like this, means not complete. There is just a mark which we can see. So that means almost the protoplasm is continuous in the hypha. The nuclei are freely present. So this is cenocytic condition. So this presence of multinucleate, many nuclei freely is called a cenocytic condition, and it is a septate hyphae. Now in the body of the Rhizopus, there are three kinds of hyphae which we can differentiate. So structurally all the hyphae are same, but based on their occurrence, we find three types. So there is a certain hyphae which branches like this, and we call it as the hold fast. Means it has a kind of grasp, a kind of grip on the substratum on which it is growing. So whether it may be a fruit or vegetable, or it may be a kind of food material. So this hyphae is called as hold fast, and this hold fast, it branches to form and give rise to another type of this holdfast. So like this is called as stolon. So this is called as holdfast, by which the hyphae has the kind of attachment to the substratum. Then from this hyphae arises elongated structures like this. So these elongated structures are called as sporangiophores. These are called as sporangiophores, which produce the reproductive structures. So sporangiophores and the holdfast and stolon are the three parts of the hyphae. So such kind of stolons which we can see, they continue and increase the growth of the Rhizopus. The holdfast, it has a kind of grip to the substratum, that is about this hyphae, and nuclei are freely present in the hyphae, and the protoplast is somewhat granular in appearance, and thus it contains numerous organelles, and ribosomes are there, endoplasmic reticulum is there, which is well branched, and there is glycogen, and what are called as the oil globules are also present in this hyphae. So this is seen, but actually when we see the hyphae, suppose this is the hyphae, the nuclei are present in the peripheral part. In the center there is a vacuole, and this vacuole which is present in the older hyphae, the vacuoles are clear, but in the tip of the hyphae, the vacuoles are not clear like this. So you can see the vacuole present clearly in the older hyphae. Actually there are many vacuoles. Now as the hyphae grows on older and older, in the older part of the hyphae, these vacuoles, they join, they coalesce, and they form a continuous cavity or the vacuole in the hyphae. So this is the structure of the thallus which is present. So the thallus of Rhizopus is of the three parts: stolons, holdfast, and sporangiophores, and the cell wall as it is made up of chitin but not cellulose. So chitin is specific fungal cellulose which is different from the animal chitin, and this cell wall is not having any kind of cellulose. Then if you come to the reproduction of the fungus, so that is about the structure of the thallus. So when we come to the reproduction, again we come across the vegetative type of reproduction, then asexual type of reproduction, and sexual type of reproduction. So in asexual and sexual, the type of reproduction is by spores, and in sexual it is by gametes. Now in vegetative type of reproduction, fragmentation is one of the common method which we can see. So even in algae, there also we come across this fragmentation. So fragmentation, it is one common method in almost most of the algae and fungi, and even there are what are called as Clamedospores is another method by vegetative method, and in addition to that there is what is called as Sporulation. So these are the three methods of so this Clamedospores or Clamedospores, they are by vegetative method, and So this sporulation is actually by asexual method, by production of spores. So fragmentation in case of vegetative, it is suppression of a particular segment of hyphae. So that fragment is capable of producing a new thallus, an independent thallus that is commonly seen in almost all algae and also fungi. So in fungi also we come across the same type of the reproduction. Then Clamedospores. So Clamedospores are one type of spores which are actually thick-walled cells. Now these Clamedospores are formed in this hyphae. So these Clamedospores which are formed, suppose this is a Clamedospore, these Clamedospores, they contain many nuclei. So the Clamedospores are thick-walled. So whenever there is unfavorable conditions for the hyphae, suppose when the hyphae is growing, if the food material is completely disappeared, means it is completely used up, consumed, when there is no more, so the hypha or this thallus will not survive due to the non-availability of the food material. So in such unfavorable conditions, so what happens? Such kind of Clamedospores are formed. So the Clamedospores are very thick-walled. They are resistant to the desiccation condition. So and they can withstand the desiccation, and these spores they can survive. So by the formation of the Clamedospore, the remaining hyphae will get destroyed due to unfavorable conditions. So when the hyphae get destroyed, the Clamedospores, they remain and they can overcome the unfavorable conditions. So and they contain sufficient reserve food material, and they are also resistant to the dry condition. So they survive and they germinate on the oncoming suitable conditions. So when the favorable conditions they means they are arrived, so there the Clamedospores will germinate into this thallus. So in case of the another method of reproduction, so that is case of sporulation, where the hyphae takes place. So this sporulation means by the production of spores. So this hyphae produces a kind of structures which are called as sporangia. So this sporangia, they are produced in any part of the hyphae. So these sporangia, they produce spores, and these sporangia, they are spherical structures which are produced at any hyphae. So now any part of the hyphae develops into sporangia. Suppose this is the hyphae, so it develops into this structure, and this is called as the sporangiophore. So this is the sporangiophore, and it gives rise to a round structure called as the sporangium. So this is the sporangium which is produced by the hyphae. So the sporangium is a spherical structure. It is usually white in color. Now as the spores are formed inside the sporangium, so when the spores are formed here, now these spores, they are multinucleate actually. So these spores appear to be black in color, and because of this sooty nature of the spores, so these sporangia appears black in color. So initially when the spores are not matured, the sporangium is white in color. Gradually it appears to be black in color. So the spores are multinucleate. So they contain numerous nuclei, and then so these spores, they are resistant to the outer conditions. They are non-motile, they don't have any flagella, and here when the sporangium is formed, inside there is a round structure produced in the sporangium, which is also the hyphae, and that is called as columella. So columella is a swollen structure inside the sporangium. So around the columella the spores are present, and which are black in color. So when the spores are completely matured, so a kind of pressure is developed in the sporangiophore and the columella. So there is a liquid accumulating in that region. So due to the accumulation of the fluid and the pressure developed in the sporangium or the sporangiophore, so the wall of the sporangium will break, and so when the wall of the sporangium breaks, so thus the the sporangium will release the spores into the external conditions. So this the spores, which are called as the sporangiospores. So sporangiospores means they are released from the sporangium. So these sporangiospores are released, and they are carried to the in different directions by the wind, or even by if there is water. And when they fall on a suitable medium, so the spores produce a kind of tube-like structure. But actually the spore germination takes place by three favorable conditions. Means three favorable conditions are necessary for the germination. So there should be adequate moisture and favorable temperature, and there should be starchy matter in the substratum. So that means carbohydrate for the germination. So starchy matter, then the suitable temperature, and also moisture is necessary. So adequate moisture is necessary for the germination of the spores. So these spores, which are multinucleate, they are having many nuclei, so they germinate and gives rise to the mycelium. So the nuclei will further divide, and this germ tube will go on developing, and as the germ tube goes on developing, so this branches and produces the mycelium. So that is how the germ tube which produces by the spore, it gives rise to a new thallus or the new mycelium with the help of the spores. This is by sporulation, production of the spores in the sporangium. So the sporangium is developed from the hyphae, fertile hyphae, which gives rise to the sporangiophore and the sporangium, and inside the sporangium there is a swollen structure called as the columella, around which the spores are present. So that is about this asexual reproduction or the sporulation. Then coming to the sexual reproduction. So sexual reproduction, of course here it is somewhat a primitive type. So we cannot mention that it is a isogamous or anisogamous or oogamous. So it is a simple type of this reproduction here. And here we come across plus and minus type of hyphae which involve in reproduction. So instead of using the word male and female, so the word plus and minus strains are used, which are opposite in nature, and so they involve in reproduction. So these two strains, plus and minus, whenever they are present, they involve in reproduction. So this sexual reproduction may be homothallic or heterothallic. So that means within the same mycelium there may be plus and minus type within the same mycelium. There are certain branches of plus type there are certain of minus type, homothallic. Now if the plus and minus strain are belonging to different thalli, so it is heterothallic. So in Rhizopus, some are homothallic and some are heterothallic. And here what happens, suppose this is a plus strain Hyphae. So this plus strain Hyphae and also minus strain Hyphae, it produces a protrusion like this. Similarly, the minus strain Hyphae also produces a protrusion in this way. So this protuberance which is produced by the Hyphae, so this develops. So it is a branch of the Hyphae, a specific branch which gives rise to the structure called as the gametangium. So this branches which are given rise by the plus type of Hyphae and minus type of Hyphae, they come in contact with each other. Suppose this is the one, and here this is another type of Hyphae. This is plus type and this is minus type. So these two come in contact with each other. So these branches which come in contact with each other, they are called as progametangia. They are called as progametangia. So the progametangia, they are the branch of a Hyphae. It is a hyphal branch. So the hyphal branch, it produces a special type of Hyphae. So that Hyphae which gives rise to the gametangia, it is actually called as zygophore. The branch which gives rise to the progametangia, zygophore. As there is a sporangiophore here, so these are called as the zygophores. The zygophore produces progametangia which come in contact with each other. So only the progametangia of plus type and minus type, they come in contact with each other, and these progametangia further go on developing. And here, as usual, the progametangia contains many nuclei. Here they are also containing stenocytic condition followed, and ultimately what happens, so the progametangia separates to form a gametangia like this. So this is the gametangia, which also contains many nuclei, and this is the part of the other Hyphae or the zygospore, which is now called as the suspensor. This is called as the suspensor, and this is the gametangium. So the progametangium develops a wall and gets separated. So that wall gets separated and forms the gametangia. So the plus gametangia and the minus gametangia. So they are formed by the progametangia by the formation of a wall here. And each gametangium contains nuclei, many nuclei. It is also multinucleate in condition. And this gametangia comes in contact with each other. So these two gametangia of plus and minus type, they fuse each other, and actually each gametangia forms a multinucleate structure which is called as the Planospores. So they are called as the planogametes, not spores. Planogametes they are called as. So each gametangium forms a planogamete. A planogamete means it is a non-motile gamete. So the protoplast of the gametangium develops into a planogamete of plus type and minus type. So the planogametes of the plus and minus types, they fuse, and this fused structure which is formed here. So this is the fused structure of the gametangium like this. So it is called as the prozygosporangium. It is called as the prozygosporangium. So the prozygosporangium, which is formed by the fusion of the aplanogametes of plus type and minus type. So this is the plus type and this is the minus type. So these two gametangia which forms the gametes, which are called as the aplanogametes, they fuse and form a structure called as prozygosporangium, which is nothing but like a zygote. So the prozygosporangium which is formed by the fusion of the two, here actually what happens, the nucleus of the plus and the minus, they fuse each other. Now suppose there are 10 nuclei here, just for example, assume here also there are 10. So the each nucleus of plus type and minus type will fuse and form a single nucleus. So the if the plus type is having 10 nuclei, if the minus is having 10 nuclei, if you assume, the prozygosporangium will also be having 10 nuclei, because the plus type nucleus and the minus type nucleus, they fuse each other. So the plus and minus type fuse each other to form a single fused nucleus, and this fused nucleus is present in the prozygosporangium. So this nucleus that belongs to the opposite strains. So after they fuse, this combined protoplast, the protoplast of the two aplanogametes, they are fused there. So they contain diploid nuclei. So these diploid nuclei which are formed, so they that structure which is formed is called as the zygospore. It forms a thick wall. So the prozygosporangium forms a thick wall there, and now this thick walled prozygosporangium, where the nucleus becomes fused, is now called as zygospore. Now soon after the fusion, the nuclei are free actually, that is called as prozygosporangium. Now when the nuclei fuse of plus and minus strains and a thick wall is formed, we call it as the zygospore. Now the zygospore is now free, it becomes separated from this thallus. The zygospore gets separated. This is the zygospore, it becomes free, that means it gets released. Now it has been attached to the suspensor of the two plus and minus strains. Now the zygospore becomes separated and it is free, and when it is set free, it undergoes a period of rest. So that means suddenly it will not develop into the next stage. So when suitable conditions arrive, there the germination of the this zygospore takes place. Now initially the zygospore undergoes meiosis. So after meiosis, the number of the nuclei will increase. They become haploid, and the haploid nuclei are freely present in the hyphae. So this hyphae now contains, or the zygospore which grows out into this vertical hyphae called germ tube or promycelium. So this zygospore will grow like this into hyphae, vertical hyphae, not horizontal. And this zygospore, which is having many nuclei, that vertical hyphae gives rise to a sporangium called as the germ sporangium. This is called as the germ sporangium. Follow. So the germ sporangium which is formed. Now actually this is called as a promycelium. This is not the real mycelium. So it gives rise to a germ tube called as a promycelium. The promycelium gives rise to the germ sporangium. So the germ sporangium, it will divide and it will give rise to spores. Now these spores which are produced here, these spores which are produced, they are called as the germ spores. So the germ spores here which are produced by the germ sporangium, they are released. They are either of plus type or minus type, or even they are both type of spores are there. So these spores are of only one type. They may be plus type, or they may be minus type, or they may be even both plus and minus type, and thus the spores are released and they give rise to a new mycelium. Now sometimes the gamtangia which are formed, they fail to fuse, means they are unable to fuse. Such gamtangium, it functions like a structure called as parthenospore, means when the gamtangia does not fuse with its opposite. Suppose if there are plus and plus type, so there is no such kind of fusion. So there the gamtangia which is unable to fuse, it forms a structure called as parthenospore, and this parthenospore develops into a thick-walled zygospore. And that thick-walled zygospore, it is just like another structure which is called as a zygospore, and it will later develop into a new mycelium. So that is how the sexual reproduction takes place, by plus and minus strain producing the zygophore, progamtangia, gamtangia, planogametes, fusion to form the prozygosporangium, then the zygospore. Zygospore produces the germ sporangium, germ spores of both or one type of spores, and these germ spores, they further develop into an independent mycelium. So that is about the life history of Rhizopus stolonifer. So in this, the structure of the thallus and the reproduction, vegetative reproduction, asexual reproduction, and sexual reproduction. So this, in case of the reproduction of this Rhizopus, actually the vegetative method is also considered as asexual method in general terms. So strictly when we take, this is vegetative method, but asexual method includes vegetative method and sporulation. Vegetative is of two types. Sporulation is another type of asexual reproduction. So this is the reproduction in Rhizopus, where the Rhizopus divides and the spores are released and carried by wind in all directions, and when they come in contact with a suitable medium, so they germinate and they develop into a new mycelium. So spores are scattered in all directions. So even spores are present everywhere, in every part of the house, in every part of the environment. So whenever we keep a food, a fruit or vegetable, so there we can see the growth of Rhizopus. There we can see the growth of the cottony mass of the hyphae or the mycelium, and this is because the spores which are released in different directions, when they come in contact with a suitable medium, they germinate and gives rise to a new kind of this hyphae or the mycelium, and that is how the reproduction takes place in this Rhizopus.